Alright guys, so moving right along in our evolution unit, we are now going to cover phylogeny and taxonomy. Hope you guys didn't have any problems with the history of life packet. That's just kind of, to me, one of the best ways to cover that, since it's a lot of hypotheticals. And so hopefully you had no issues with the web quest or answering any of those questions. So we're going to continue on, and this lesson is going to be about phylogeny and taxonomy. And you may or may not remember those words from freshman year. And so we're just going to start off just a little bit of definitions, but phylogeny is going to be the evolutionary history of a species. Now you do have to keep in mind that in biology, everything descended from common ancestors, okay? And so we pretty much reconstruct this phylogeny of, where did these organisms come from using systematics. And so this particular lesson is actually going to kind of go over some of the same things you looked at in that evolution evidence packet that you did at the very beginning. And so we're going to take a look at some different areas of study that help really evolutionists and biologists kind of recreate these evolutionary histories. And so one major way is through the study of fossils. And essentially we kind of have this timeline in rocks, because we know that sedimentary rocks are layered, and the deeper the layer, the older they are. And so by finding fossils in these various layers, we can kind of create what came first, what came second, and so forth. So the deeper they are, the older they are. And this is what we call relative dating. Relative is dating them based on the layers that they are in those sedimentary rocks. Now, absolute dating is where they bring in pretty much some math and some chemistry, and you look at radioisotopes and half-lives, and you can look at ratios, because as certain radioactive isotopes decay, they turn into other elements. So you can look at these ratios, apply some math based on their half-lives, and you're able to get a little bit more of an accurate timeline of exactly how far back that fossil is dated. And so between the two, relative dating and absolute dating, we can essentially kind of reconstruct life history. Now, obviously, there are some criticisms with this, and that not everything is going to be fossilized. Okay, a lot of soft-bodied organisms are not going to be found in the fossil record. It really just depends on the conditions at the time, and hopefully you learned a little bit about fossils in your evidence lab. But at least we do have several different organisms and species that are found in the fossil record. So even though it's not complete, it is quite substantial. Another area of evidence is based on geology, and kind of goes along with that biogeography that you looked at, and that, as the Earth changes, we know that that can affect, geographic distribution. So if you look at geologic changes like, continental drift, mountain building, things of that nature, as the Earth changes, so does the biogeography. And so hopefully you probably learned somewhere back in middle school, I would imagine, about Pangaea and the supercontinent, and how that they have split over time, and just this change in geographic location changes could lead to these different evolutionary episodes, mass extinctions, and so just the constant idea of continental drift, that the continents are moving. Another thing that's studied are these mass extinctions, and this is something that you had in your history of life packet. There are believed to be 5 mass extinctions, and we are currently, they believe, experiencing the 6th, which is human-related. But these mass extinctions are represented in the fossil record, where you see kind of an abrupt change. Back when we covered some evolutionary mechanisms, we looked at timing, and we looked at gradualism versus punctuated equilibrium. And what you find in the fossil record is that after these mass extinctions, you usually kind of had rapid change. And so mass extinctions, when several organisms or the majority of organisms are wiped out, it leads to kind of new habitats, new resources for those survivors, and they're able to kind of diversify in order to continue to survive. So here's just that continental drift concept. Here you have Pangaea. You can see about 225 million years ago, and all the way up to the present. And this is probably an old picture. They're continuing to move today. But we do know that these continents have shifted over time, and that can affect distribution of organisms. All right, another major area of study is homologous structures, okay? And this is something I want to take a minute to discuss because back in your evolutionary evidence lab that you did, it covered both homologous structures and analogous structures. Now, in terms of determining relatedness, the only structures that would be helpful are those that are homologous because essentially homologous structures are structures that are similar because there is a relationship, okay? Now, analogous structures do not necessarily show a relationship because that has to do with environmental pressures. Analogous structures arise from convergent evolution when organisms have similar structures, not because they're related, but because they're living in the same places, they're under the same pressures. So I just want to be clear that these are not going to be helpful. Just because two things look alike, think about that dolphin and that shark, it does not necessarily mean that they're related. That dolphin is more related to you and me than it is that shark, okay? So in order to determine relationships in evolutionary history, we have to look at homologous structures. And one of the most common examples are these vertebrate limbs. So we know that the function of these are different, okay? Our hands and arms have a completely different function than a dolphin flipper or a bat wing. But yet if you look at that bone structure, and you were supposed to color this in your evidence lab, if you look at that bone structure, you can see that it's very similar. You have the same basic two bone structure in kind of like the forearm area. You have the carpals, the metacarpals, the phalanges. That same basic bone structure exists in all of these limbs, even though they don't necessarily do the same thing. So that does indicate a relationship amongst these organisms, okay? Now, probably one of the most widely used areas for evidence today is molecular data, comparing DNA sequences and protein sequences. And we can kind of get it down to the letter, like literally we can get it down to the letter of how many differences in nitrogen bases are there. We kind of saw that in the ice fish, how mutations can arise. And so looking at molecular data, they can determine how closely two species are related. Now, another branch of science is called taxonomy, and taxonomy is like the actual classifying and naming branch. It was started a long time ago, back with Carolus Linnaeus, and this is at a time when Latin is the primary language. And so every organism on the planet that has ever been discovered is going to be given a scientific name, and it's going to be classified in our taxonomic scheme. And so in terms of the naming system, it's a two-part naming system known as binomial nomenclature. This literally means two-name naming system. And so every organism has a scientific name. This is a universal language because obviously we don't speak the same language across the globe. Words like cat in English and Spanish and French, it's going to be different, okay? It's not cat in every language. And so we want to have this universal language in science, and for organisms, that's going to be binomial nomenclature. And so that Felis domesticus, that's your basic house cat. So it doesn't matter what country you're in or what language is spoken, a house cat is always going to be Felis domesticus. Our scientific name is Homo sapiens. A dog is Canis familiaris. This Drosophila melanogaster here, that is your fruit flies that we studied in genetics. And so they always have the same basic format, okay? It's two parts. The first part is always the genus that organism fits into, as well as their species name. And so the species is the lowest level that you can get. The genus is always capitalized, the species is lowercase, and then the scientific name is italicized. So if you ever see those kind of funny looking words, it's most likely going to be your scientific name. And so the basic hierarchy, or taxonomic scheme, is a hierarchy from very, very broad all the way down to very specific. Hopefully you kind of remember this back from freshman year. There's some mnemonic devices like Dashing King Philip came over from Great Spain, or Daring King Philip came over for grape soda, or something like that. But essentially this is, the classification scheme that we use. And so every organism, once discovered, is going to hopefully be placed into these categories. And I'll be honest with you, it can be kind of difficult. This is one of those things that in science, taxonomy is always changing, because as we learn more, and as we're able to study DNA sequences, remember that just because two things look alike doesn't mean they're related. We kind of have to look at several different areas to determine how these organisms are going to be classified. And so the most broad category is the domain. And then from domain, you go down into kingdoms. And we're going to as we finish out the year, we're going to go into the various kingdoms and the groups. We'll talk about the domains individually. From kingdom, it's to phylum, then class, order, family, genus, species. And in my opinion, this is one of those things that you should just know, you should have memorized. So domain, kingdom, phylum, class, order, family, genus, species. So each level is a taxon, or plural is taxa. And the lower the level that you share, the more related you are. So for example, humans are in domain Eukarya. We are in kingdom Animalia, phylum Chordata, class Mammalia, order Primates, family Hominidae, genus Homo, and species Sapiens. And so I don't know that we have any other families in Hominidae that are currently still alive, but we are in the same order as other primates, okay? We are in the same class as all mammals, so obviously I'm going to be more related to something that's in the order primates than I am just a mammal like an elephant, okay? And then we are all in kingdom Animalia, so I am related to other animals like an earthworm or a butterfly, but I'm going to be related to mammals than I would be just say something that I share the same kingdom with. So the lower the level that you share, the more closely related two organisms are. You can't get more related than a species. We saw that with that biological species concept, that interbreeding group that produces viable offspring. So the next closest related you can have is the same genus, then after that would be family and so forth. And so here's one for the panther, okay, because kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Phylogeny, genus Panthera, and species Pardus. So that together is going to give you that scientific name. Okay, so once again, it's going from very, very broad all the way down to very specific. All right, so we have three domains that we group organisms into. And honestly, I don't really know how long domains have been around. I don't really know about them until I started teaching. I don't remember learning these in school. And like I mentioned, taxonomy is always changing. Back when I was in school, we learned a 5-kingdom system. Freshman year, you learned a 6-kingdom system. And the reality is, I don't even know the official number of kingdoms currently because they constantly change it. But bacteria is going to be the domain that includes common everyday bacteria. And one of the key definers for domain bacteria, I did the wrong thing there, is peptidoglycan. Okay, so we're going to learn more about this when we get to bacteria. But peptidoglycan is the substance that makes up the cell walls for this group, okay? Now, notice that these are bacteria, so they're going to be prokaryotes, so this is a prokaryotic domain, but one of the things that sets them apart is that peptidoglycan, because we do have another prokaryotic domain, okay? In fact, this right here probably needs to be changed. They're actually not even called bacteria, they're called archaeans, or it really should say extreme prokaryotes that lack peptidoglycan. And so these guys are kind of some extremophiles. They're kind of weird. They live in places that a lot of other living things would not be able to survive and thrive in. They can live in really hot temperatures. They can live in very acidic pHs or extreme pH, extreme salinity. And so they are put into kingdom Archaea. So really they're not even called bacteria. So we have bacteria which have pep, and we have archaeans that don't, okay? These are your kind of common everyday, the ones that might make us sick, like, you know, streptococcus or staphylococcus, E. coli, things like that, good and bad. And then we have the archaeans, which are going to live in those extreme environments. And then everything else, all of your eukaryotes are going to be in domain Eukarya. So we have kingdom fungi, Plantae, and Animalia. Protists used to be a kingdom called Protista, but it's since been divided. And like I mentioned, I actually don't even know how many kingdoms there are that came out of kingdom Protista. All right, so how do we kind of represent these relationships? So if we're trying to trace back these ancestral family trees, we do that using phylogenetic trees and cladograms. So the purpose of these particular models are to kind of reconstruct evolutionary history. And I do want to point out that they are hypothetical, okay? The reality is we'll never know for certain exactly where every branch happened and what organisms came from exactly what. When you're talking, you know, 1 billion years ago, hundreds of thousands of years ago, it's going to be almost impossible to get down these specifics. but it can at least kind of help us recreate these evolutionary relationships. So it does represent the divergence of species from a common ancestor, and typically, sometimes with a cladogram especially, it shows the emergence of innovative characteristics or new traits that you'll find within groups. And so it's a series of branches, okay? And so here's an example of a phylogenetic tree. Every time there's a branch, we call that a node, okay? So this is a node, this is a node, this is a node. And what the nodes do is they represent common ancestors. So this would be the common ancestor to essentially all life. This is a very broad phylogenetic tree that has all three of our domains on it. One thing I want to point out here is notice this ancestor here, archaeans, which are prokaryotes, appear to be more closely related to eukarya than they do bacteria, because you can see that they're coming off the same branch. So the closer the branch that they share, the more related they are, okay? So like your diplomonads would be more related to Microsporidia than, say, the ciliates, okay? So each branch represents a node or a common ancestor, and the closer the node, the more related they are. Now here's an example of a cladogram. This is a very basic one, okay? And this is one that shows that evolutionary relationship based on innovative characteristics. So technically down here would be your precursor, your ancestor to all vertebrates, because from the hagfish to the chimp, these are all organisms that have a backbone, okay? The length of the line represents time, okay? So the shorter the line, the shorter they've been around, the longer the line, the further back they go in time. And you can see that as new traits emerge, we see these new relationships. And just like before, every branch is a node, and every node represents a common ancestor. So here we have the hagfish, and you can see by these lines here that they do not have things like jaws or lungs or claws or nails, okay? But the innovative characteristic here would be jaws, and so after that, all of your vertebrates have jaws. So these are called your bony fish, okay? Fish do not have lungs, so notice that comes after the fish, but amphibians do, reptiles do, and so forth. Sometimes you'll see some specific traits on a single branch, so like feathers would not be down here on the main line because they don't all have feathers, so it's specific to those birds. But this is just showing the emergence of new traits and how organisms kind of evolve over time. And then there's another phylogenetic tree that kind of represents all life, the same kind of basic branches. You have true bacteria, archaebacteria, eukaryotes, and then it branches right on down. So when we're looking at these cladograms, those that have like these innovative characteristics, okay, each evolutionary branch is called a clade. And the clades can be described as monophyletic, paraphyletic, or polyphyletic. Now the most accepted group is a monophyletic group because this has pretty much the ancestor, which is going to be that node, and all of its descendants. So this right here would be a monophyletic clade, okay? So it has the descendant and it has all of the ancestors. Now some other groupings that you might see describing a group of organisms would be paraphyletic. So this is where you have the ancestor, but you're not including all of the descendants, so this would be a paraphyletic group. And then polyphyletic is where you have a group that you're referring to, but you're not including that common ancestor, so you can see that that ancestor is excluded there. These are not necessarily considered clades, but when you're describing evolutionary relationships or groups of organisms, you could use these words to describe them. Now, how do they determine the sequence? How do they determine what comes first, what comes next? Well, like I told you earlier, it's really complicated, and this is one of the reasons why these phylogenetic trees are going to be hypothetical, because there's a lot of different things that you can look at to determine relatedness. You can look at, the fossil records, you can look at genetic sequences, you can look at common traits. And so one thing they do look at is those homologous structures. So the more homologies that they share, the more closely related they're going to be, okay? You can look at those shared characters, like for instance, mammals and hair, or it could be reptiles and scales, or it could be the amphibians and the slimy skin with lungs, okay? And then you also normally have an out-group comparison. So that's going to be something that's just outside of that group. So earlier when I showed you, I'm going to go back, So earlier when I showed you, this is a vertebrate cladogram. Okay, so your outgroup comparison would be something that is not a vertebrate. Okay, it would actually probably be an invertebrate chordate, which we'll learn about later. But you're going to have an outgroup. So if we were doing a comparison of mammals, your outgroup might be a reptile. Okay, and so typically you're going to have something that's just outside of that grouping that you can compare them to. All right, now I mentioned earlier that one of the most commonly used tools today is going to be DNA. And so there's a field known as bioinformatics where you analyze DNA. It involves a lot of computer work, but essentially you can put in DNA sequences. It allows us to try to determine the functions of proteins, and it allows us to compare the genomes of different organisms. Okay, once again, the more similarities they have, the more related they are going to be. And so we commonly use these nucleotide sequences or these protein sequences to construct these cladograms. And so what they do is they kind of use the concept of mutations and how quickly those mutations occur and how quickly they can add up to kind of determine rates of change. So there are some particular sequences like rRNA. It pretty much changes slowly over time. This is something that's going to be similar for the majority of organisms on the planet. It's relatively steady and mutates very slowly, whereas something like mitochondrial DNA, which is that DNA specifically in the mitochondria, it gets mutations very rapidly. And so if they're trying to compare organisms that are more recently linked, they might compare their mitochondrial DNA sequences. If they're trying to compare organisms that are more distantly linked, they may look at rRNA sequences. And so they can also tie in what we call molecular clocks. Okay, so essentially, I'm going to simplify this because I'm sure it's much more complicated, but it's applying these mutation rates to how far back the ancestor may go. Okay, so you take the number of mutations essentially, and that's proportional to the time since those two groups diverged. Okay. So if you're using, let's say, a mutation rate that's rather quick, let's say we get, 2 mutations every 1,000 years or something like that, then if there were 6 mutations between the two groups, that would take us back, 3,000 years. And so all you do is you just apply a basic ratio for mutation rates, how many mutations there were, and that can kind of take you back to how long ago those two diverged into new species. Okay. All right. Now, as I mentioned earlier, this can be difficult. Okay, because there's a lot of factors. What if they look alike? What if they have a lot of homologies? What if their DNA is similar? Or what if their DNA is different, but they have a lot of physical homologies? Well, how do you determine the best case scenario? Pretty much keep it simple, stupid. Okay, you've probably heard that phrase before, or KISS. But we're just going to keep it simple, which is the principle of parsimony. And so essentially the most logical, simplest explanation is probably going to be the best. As I mentioned, this is going to be hypothetical, right? This is not something that we will ever know with certainty. We could also use the principle of Occam's razor to shave off unnecessary complications. And so once again, we're just going to go with what appears to be the most logical explanation. And really in today's world, it's going to be a lot about those DNA sequences. So that wraps up this lesson on phylogeny and taxonomy. I hope that makes some sense to you. Over the course of the next week or so, you guys are going to be continuing to watch videos. We still have a few more lessons. Hopefully I'll be around this week, and I will talk to you then. Hope you guys have a great day.